Hi, this is Samantha Mollo, and this is my oral defense for EVSP 699 Capstone Proposal Thesis on December 22nd, 2019. The title of my thesis is Stubborn Pharmaceuticals and Personal Care Products, Proficient Removal Methods of Wastewater Treatment Facilities. The background of this problem includes the reasoning that pharmaceuticals and personal care products are suspected to being very harmful to the aquatic environment. They're a modern problem that has been detected within the last 15 years, and their effects are being studied on wildlife, aquatic ecosystems, and humans. These effects virtually are mostly unknown right now. There are a few things that we do know. We do know that it causes endocrine disruption in fish such as rainbow trout and carp. And we also know that PPCPs are very common. They're things found in the drugs of all forms that we use. They're the chemicals within toiletries, cleaning agents, sunscreens. They're used in agricultural using, especially for irrigation and such, fragrances, and they're entered the waterways by human wastes when we take a drug and then we have our wastes. It re-enters the water system. There's also agricultural production, drug removal from flushing down the toilet, and it creates PPCPs in both lotic and lentic waters. Wastewater treatment centers contribute their own PPCPs because they started using chloramine to disinfect drinking water, and this creates nitrosamines, so that's also adding to the problem. The issue is that many of the studies have shown that PPCPs aren't eradicated after treatment, and they re-enter the drinking water as water is a renewable resource. So this increases their concentration as water is renewing and renewing. And what happens is it starts to affect the wildlife that's in aquatic ecosystems, but it's also can have detrimental effects to the people drinking the drinking water. Even when PPCPs were brought to tolerant levels in a study in 2014, rainbow trout were shown to still have endocrine disruption. So their long-term effects could be more disastrous than we actually know currently. With them being so prevalent in modern times, it's really important to find out which methods remove the most amount, and then which are the PPCPs that are not being removed, which are the ones that are being concentrated more and more as more water is entering each system. So the research questions that we have, or I have, is which removal method of pharmaceuticals and personal care products is the most efficient? Which one really does the best job? And then from there, which PPCPs are the hardest to remove? And then what recommendations are there so we can remove them in the future in some kind of capacity? The justification for these questions is that even through biodegradation, photodegradation, hydrolysis, sorption, activated carbon absorption, ozonation, oxidation, membrane separation, there are a few still leaking through that are not breaking down correctly. carbamazepine and sulfamethoxazole seem to be the hardest ones that have shown in the studies to be eradicated. And we need a comparative study to show how this can be economical and work, hopefully. So the thesis is novel magnetic carboxyl modified hypercross-linked resins seem to be have the best adsorption capabilities because of their polarity, their pores, and they have a really nice large surface area. Other treatments that have been promising are aerobic granular sludge membrane bioreactors, hydroxyapatite GAC adsorption, um, but still these sulfamethoxazole and carbamazepine still have a large concentration. So is there a way that we can compare them to uh, other structures or that have been broken down and recommend something such as silver reduced graphene oxide nanocomposite? So we'll see. Uh, the data sources that we'll be using is mostly from the American Public University Systems Library, a little bit from Google Scholar, but I made sure that all of them are peer-reviewed journals and they're published less than 10 years ago. And since this is such a current issue, it's been fine. The analytical procedures that we'll be using are we'll have treatment such as the ones mentioned before, and then the PPCPs, all the ones listed here, caffeine, acetaminophen, on and on. And the statistical information of how each of these treatments break down each of these PPCPs will be listed and show which treatment breaks down the most of them all at once. And maybe we'll have to integrate 2 treatments or more to have the best answer of how we can get rid of PPCPs. But which one singular also is going to really give you the most breaking down. Then the most stubborn PPCPs will be examined additionally and be provided with a little bit of recommendation for other researchers to explore how we can break these down better. By expanding treatment knowledge through comparison and possibly integrating a couple of treatment methods, removal can be more transparent, and then the community can kind of understand which PPCPs need more attention and can hopefully give more information about how these can be eradicated. And these are the references from my slides, and I appreciate you taking the time to listen. Thank you.